for our records, but also for your electronic certificate that we will send to your email. For your information, the certificate both days of the workshop. Thank you for joining the workshop today, since yesterday. This is actually the first of the 3 online workshops that the COSMARINDO program is organizing this year. And for you who just joined us today, I will explain again a little bit about this program. So COSMARINDO is a program that aims to improve the coral restoration efforts in Indonesia by using larval propagation.
Menggunakan perkembangbiakan.
Are funded by Korda, the Coral Research and Development. The program itself is put together by the team from Ruhr University Bochum of the Ryukyus Japan and Unseen France. We are collaborating with Indonesian universities, Universitas Banda Neira, Universitas Patimura. They are both our collaborators for a long time now. And then we have Universitas Hasanuddin, Universitas Mataram, Universitas Samratulangi, and Universitas Udayana. So yesterday with Dr. Joanna Diaz and Dr. Marijke Hun, we learned about the different strategies that corals use to reproduce, and also the environmental cues that influence the timing of their reproductive events. Today, we will have 3 speakers who will present 2 topics. There is a change in the presentation order. So first, we will have Dr. Frédéric Sinige from the University of the Ryukyus, Japan, and Alexis Chapuis from Unseen France, who will bring us to dive deeper to introduce us to the mesophotic coral ecosystems. And after that, we will have Professor Saki Hari from...
Other way around. Starting with Saki.
Sorry, yeah. We will have Professor Saki Hari from the University of the Ryukyus, Japan, who will speak about the biology of coral larvae settlement, and early life of coral liquids. And then on the second presentation, we will have first Dr. Frédéric Sinige from the University of the Ryukyus, Japan, and Alexis Chapuis from Unseen France. And after each presentation, we will have sessions. You can either type your questions in the chat box or raise your hand, and we will unmute you to have a direct interaction with our speakers. Please note that each discussion session will be limited to 20 minutes only. So again, don't forget to please fill the attendance form by clicking the link provided in the chat box. Let's begin this workshop with a presentation from Professor Saki Hari about the biology of coral larvae settlement, and early life of coral liquids. Professor Saki Hari, the time is yours.
Thank you very much for your introduction. And I will share the screen. Please wait. Is it good? Connected? Can you see? And can you hear me well?
Yes.
Okay, good. Selamat siang, and everyone. I'm Saki Hari, a marine biologist studying coral reefs at University of the Ryukyus in Okinawa, Japan. Coral and Marquez coral including larvae and juveniles. Before explaining the main topics, let me introduce our marine research station in Okinawa. The station faces nice coral reefs and welcomes both domestic and international students and collaborators. We have a dormitory here, allowing visitors to stay and work in the station. The station is just over 50 years old, but the dormitory is in a new building. Over the 50 years, Many contributions to coral science have been made in the station, including the sex change research at the journal presented yesterday about the fungit. Continue. So, we have a small boat for field work around the island, and then we also have running seawater systems, allowing us to keep marine organisms, including corals. As you can see, we have nice coral reefs around the island on your right picture. Unfortunately, they are damaged by the bleaching last year, but we hope that they will recover soon. If you are interested in learning about coral reefs in Okinawa, our university provides scholarships for master's and doctoral courses, so feel free to contact us for future information. So now, let's move on to today's topic. As Joanna and Marika explained yesterday,
Seperti yang dijelaskan oleh
corals normally spawn at night, and eggs develop into the plant larvae like that. They disperse several days and several 10 days in the ocean before settlement. Some corals are brooders, which release larvae directly into the water. The larvae can swim using cilia, kind of many short hairs on larvae, and go down to the sea bottom to attach the substrate. We call this settlement. After settlement, they metamorph into polyps. And they start to making skeletons and grow asexually and become member of the reefs. At this point, we are called recruits. I quickly show you a picture from spawning to settlement. I will not show you the spawning video because Joanna and Marika showed very well yesterday. And this picture shows coral spawning at night. You can see the bundles floating on the surface. But a bundle looks like an egg, but it actually contains several eggs and sperm. Since many corals spawn during the same night, sometimes you can see egg aggregation on the sea surface following the morning, like that. We call it coral slick. This picture shows the eggs development after spawning. We can see cell divisions starting for the 2 cells and the 4 cells from 2 hours on the top left picture and to about 9 hours on the right. The bottom right shows 2 days later. We call it embryo. Finally, 4 days after spawning, the egg becomes a fully developed planular larva. At this stage, they start settling. Here, I show you a video of coral larvae. They are 4 to 5 days old and less than 1 millimeter long. As you can see, they swim actively. The larvae swim with the side opposite to their mouth, leading the way. So they kind of swim backward. As you can imagine, they cannot swim against strong currents, but can change their positions in the water column and search for their settlement place. After reaching the bottom, they start to crawl to find a place to settlement. Normally, larvae start settling from 3 to 4 days after spawning for spawners, and soon after releasing for brooders. They can extend their planktonic periods to more than a month for acropolar species. Once the larvae settle, they cannot move to another place, so it is crucial for them to select very well. There are many studies on the settlement queue. For acropola, the coralline algae, shown in the pink part in this picture, are considered one of the settlement queue. Other studies show that the bacteria community on the algae is important for settlement. So after the settlement, they metamorph into a tiny polyp, which is in 1 millimeter length, and they develop tentacles like a flower, and with a mouth in the center. The left picture shows the polyps 3 weeks after settlement. And inside the polyp, the brown part is symbiotic algae acquired from seawater through the mouth. You can also see the skeleton and the tissues, this part. We can call this stage polar recruit or polar spot. After 1 year, they grow many polyps and branches. The diameter at this stage is about 1 centimeter. Symbiotic organisms often live in the coral branches, as you know. In the right picture, you can see that a juvenile crab here that has come the first year after the coral settlement. And these pictures show a brooder releasing larvae. For many brooders, they release larvae monthly, except during the cold season. I think in Indonesia, they release, you can see larvae release every month, yearly. But in Okinawa, we can see it only in summer. The larvae are seen inside of the polyp in this picture. This, the dark brown part is a larva. And then released here. They normally have more chance to recruit to the reefs compared to the spawners because of multiple releasing times per year. However, they produce less larvae per time. So let's look at the dispersal more in detail. The larval dispersal means moving the larvae from spawned place to a settlement place. The larval dispersal is important for the ecologically maintaining and expanding a population, as they are sessile organisms, evolutionary, increasing genetic diversity, as Joanna explained, and species speciation. And in a conservation aspect, a recovery from the damaged reefs. A group of populations connected by larval dispersal are called metapopulations. The subpopulations maintained each other through the larval dispersal and recruitment. On the other hand, a population like D is isolated from the metapopulations. Under the environmental changes such as marine heat waves, coral often experience significant mortality like population B, C, D. Larval dispersal and recruitment is crucial for reef recovery from a healthy population, in this figure, like a population A. From A to B, A to C. However, population D has less hope for recovery as it is isolated with no larval source around. Now let's think about what would happen to sexual reproduction of corals if ocean warms. I remember yesterday, someone had the same question. Some researches showed timing of the spawning may be changed, like less synchronous, and then they are more splitted. Regarding the larval dispersal, my colleague and I published this paper, showed the changes in larval dispersal ranges. In the laboratory, the eggs and larvae were cultured under the different temperature conditions from 27 to 31 degrees in Celsius. And then we found one in the high mortality. This graph shows the survivorship. And then this one is the higher temperature condition. And it has high mortality compared to the normal condition. And two, this graph shows the settlement rate. And interestingly, the warm condition, they settle earlier than normal condition, but settlement rate is lower than the normal conditions. Together with the larval modeling, which I will not explain in detail in this presentation, but we concluded that the dispersal range will be narrower than the present condition, enhancing the self-recruitment. But fragmented metapopulations, as the larval performance is lower, which means lower survival and then lower settlement. So now let's look at the fundamental larval dispersal topic. Do you know what controls larval dispersal ranges? There are two big factors, biologically and physically. For biological factor, the following larval traits are affected. Up position in the water column by buoyancy and or larval behavior. And second one is the survivorship. And third one is settlement competency period, which are defined as the periods when larvae are able have the ability to settle. This one is also affected by their nutrient status. For physical factors, as you can easily imagine, the current is major factor controlling the larval dispersal. But in the current flow, it could be big oceanic current or wind-driven current or locally tidal current. And water temperature and light are also affected to the larval dispersal. And together, these factors determine where larvae will disperse. For example, like a coral spawn massively and the eggs are floating on the surface. In this case, they may receive wind-driven current or tidal currents. But once they develop and start to swim to the sea bottom, they may receive the different current and close to the sea bottom. So today I will go into more like biological factors. As you can see, there are many different types of the larvae from the different coral species. They have different size from the 50 micrometer to the more than 2 millimeter in length, and different symbiotic status and food reserves stored as lipid. We can see different colors of the larvae, for example, green, and more oranges, and pinkish, and white. But I don't know the meaning of those differences, except of the brown larvae shown in the bottom lines that obtain their color from the symbiotic algae. In Acropora, the eggs are usually rich in lipid, representing more than 60% of the dry weights. Because of that, the eggs float on the surface, enhancing the fertilization success. Indeed, at the surface, because it is now into the 2 dimensions instead of the 3 dimension in the water column, the eggs and sperm can encounter easier than 3D dimension. In the larval stage, they use lipid as energy source. This allows them to disperse more than a month without food. And regarding the symbiotic algae, coral are associated with unicellular dinoflagellates sometimes called zooxanthellae, but now usually refer to them as Symbiodiniaceae, because the genus Zooxanthella is a different dinoflagellate. As I told previously about the larvae, the brown color is given by the algae. And you can see on the right picture that there are a lot of algae in the coral tissues. As every little brown grain is an alga. The size of an alga is 0.01 millimeter, very tiny. The establishment of the symbiosis occurs in the early life stages of corals. There are 2 different modes of acquisition: vertical transmission or horizontal transmission. For the vertical transmission, the eggs or larvae inherit the symbiont from the parent colony. On the other hand, horizontal transmission, the eggs or larvae do not contain symbionts, and they must acquire them from the environment. The majority of corals are spawner, as Joanna explained yesterday, and most of them are the horizontal transmitter. These horizontal transmitters acquire symbiont either polyp stage or the larval stage. As you can see on the right bottom picture, under the fluorescence microscope, the larvae looks green, and the red particles here are actually the symbionts. The little star shows that they are where the mouth is, so they acquire symbiont through the mouth into the larvae. So what is the advantage of having symbionts as a larva? it is still an open, ongoing research field with different researchers having different opinions. In our research, we found that larvae with symbiont survive better. Yeah, this graph shows the survivorship of the larvae, and this shows the larvae with symbiont, and this is the without algae. And larvae with symbiont survives better than the without algae. And this graph shows the changes in lipid, and the larvae with symbiont use less lipids than the without algae, suggesting that the algae provides some energy to the larvae. However, other researchers also show that the algae can be harmful for the larvae, as they can be a source of oxidative damages under strong UV and/or high temperatures. And regarding to the Symbarinaceae, it is not a species but a family name. This family contains several genera, and each genus contains several species or types. The different types have different physiological properties, and this can affect the environmental tolerances. For example, the genus Drusidium has been known for its thermal tolerance and persists in coral after bleaching. Why does the diversity of symbiont matters at early life stages? At the beginning, they acquire many algal types. The composition of the symbiont community in juvenile polyps changes as coral grow to the becoming grows to become dominated one or few types in the adult corals. These mechanisms may allow them to adapt the local environment where they settle. Finally, I would like to show a few images of what happened in my lab during coral spawning. We normally collect the mature colonies and then transport them to running seawater aquariums where we set some bundle collectors. Of course, we get a permission to collect the corals, and we try to limit the collection to minimum for our researches. And after spawning, we return the colony to the reef. Then every night, we observe coral until they spawn. As Marike explained, the timing of the spawning, prediction of the timing of spawning is very difficult. So we just observe every night until when they spawn. So when they finally spawn, we collect the bundles and mix them between colonies to fertilize the eggs in the laboratory. We then culture the larvae in the filtered seawater, which we change daily. After we conducted different experiment, after that we conduct a different experiment in the laboratory, or we transplant the juveniles back to the reef and observe for the future experiments. On the bottom right picture, you can see this person named Angga, my Indonesian PhD student. Then during the spawning, we all work together. So sometimes we deploy the equipment tiles, which is 10 by 10 centimeter, in the reef directory. This is to know which corals recruit where. So several months later, we collect tiles and identify juvenile of the corals. Typically, we find quite diverse range of coral species on the settlement tiles. Because of that, understanding when coral spawns at which location site is crucial. So if you are interested in this, please join our coral spawning network. So, terima kasih. And thank you, Angar, for my slide text translation. And then Alan for the online translation. Thank you.
Thank you very much, Saki, for your presentation.